Hello students, today I bring for you a new chapter and that is plant growth and development. In this chapter we are going to cover many important topics. So let's start. First of all, what is plant growth or what are plants and how they grow? Okay, so all plant cells are descendant of the zygote. Means they are comes from zygote. Zygote is a fertilized egg. The zygote develops into a mature plant through growth and differentiation, forming root, leaves, branches, flowers, fruits and seeds. Then they eventually die. As you can see, zygote, 2-cell, 8-celled embryo, globular embryo, heart-shaped embryo, torpedo embryo and then mature plant. Plant growth. Growth is an irreversible permanent increase in size of an organ or its part or an individual cell. It involves metabolic process that consumes energy. Plant growth generally indeterminate means plant growth continues throughout the life due to the presence of meristems. Meristematic cells have the capacity to divide and self-perpetuate. Perpetuate means the capability of something to cause itself to continue to exist. That is called self-perpetuate. Okay. The growth where new cells are always added to the plant body by the activity of the meristem is called open form of growth. Here you can see the diagram where there are different type of meristems are present. As you can see, shoot apical meristem present in the shoot tip of young leaves, and lateral meristem, vascular cambium, cork cambium, axillary bud meristem, root apical meristem, and primary growth in stem that have epidermis, cortex, primary phloem, primary xylem, pith. Secondary growth in stem which have, you can see, periderm, cork cambium, cortex, primary phloem, secondary phloem, vascular cambium, pith, primary xylem, secondary xylem. And this is the diagram. It is also in your NCERT. Here you can see vascular cambium, shoot apical meristem, and in root portion, vascular cambium, root apical meristem. Primary growth, it occurs due to root apical meristem and shoot apical meristem. It causes the elongation of plants along their axis. Secondary growth in gymnosperms and dicots, it occurs due to lateral meristems, vascular cambium and cork cambium. These cause the increase in the girth of the organs. Girth means width of the organ. Okay. Growth is measurable. Means at cellular level, growth occurs due to increase in the amount of protoplasm. Protoplasm means the portions present inside the cell, that is cytoplasm and nucleus, combinedly called protoplasm. Increase in protoplasm is difficult to measure directly. So growth is measured by parameters like increase in fresh weight, dry weight, length, area, volume and cell number. What do you mean by fresh weight? Now fresh weight means, it is also called wet weight, means the weight of a plant or plant part measured immediately after harvesting or removal from its natural environment. That is called fresh weight. Dry weight means it refers to the weight of plant tissue or organ after it has been completely dried to remove all moisture. That is called dry weight. And you can see cell number. A maize root apical meristem can produce more than 17,500 new cells per hour. Cell size. Cells in a watermelon may increase in size by up to 350,000 times. Length, growth of a pollen tube. Surface area, growth in a dorsiventral leaf. Next is phases of growth. First of all, meristematic phase. It occurs in the meristems at the root apex and shoot apex. Apex means the top portion of root and shoot. Okay. Cell in this region have rich protoplasm and larger nuclei. Cell wall are primary, thin and cellulosic with abundant plasmodesmata. Elongation phase, It occurs in cells proximal, just next, away from the tip to meristematic zone. Cell have increased vacuolation, size and new cellular deposition. And in maturation phase, it occurs in the cells further away from the apex, more proximal to the phase of elongation. The cells attain maximal size in terms of wall thickening and protoplasmic modifications. We can see this diagram. Detection of zones of elongation by the parallel line technique. Zone ABCD immediately behind the apex have elongated most. Then EFG, they are not elongated. Now coming to growth rate. It is the increased growth per unit time. The growth rate may be arithmetic or geometrical. Arithmetic means arithmetic growth. In this, following mitotic cell division, only one daughter cell continue to divide while the other differentiate and mature. You can see the first diagram, arithmetic, only one daughter cell is dividing while the other cells, they differentiate and they do their other work. Okay. See the color combinations. In below you can see this diagram. Cells capable of division, that is blue color, and cells that lose capacity to divide, which are differentiate and mature. The color is changed. On plotting the length of the organ against time, a linear curve is obtained, like this diagram inside this. Okay, in your screen. Linear curve is obtained in arithmetic growth. Now, mathematically, it is expressed as LT equal to L naught plus RT, where LT or LT is the length at time t, L naught or L0 is the length at time 0, R is the growth rate or elongation per unit time. It is arithmetic growth. Now coming to geometrical growth. Here you can see both daughter cells continue mitotic cell division and geometric growth. In most systems, the initial growth is slow, that is called lag phase. Then it increases rapidly, that is log or exponential phase. If nutrient supply is limited, the growth slows down, leading to stationary phase. On plotting the parameter of the growth against time, we get a sigmoid or S curve. As you can see, lag phase is the initial phase, which is also called slow phase. Then coming to rapidly growth phase, that is called log phase or exponential phase. And if the nutrient supply is limited, then the growth slows down and that leads to stationary phase. So by plotting this parameter of growth against time, we get a sigmoid curve. So geometrical growth is a growth gives sigmoid curve. Okay. Now, a sigmoid curve is a characteristic of living organism growing in a natural environment. It is typical for all cells, tissue and organs of a plant. The exponential growth can be expressed as W1 equal to W0 ERT, where W1 is the final size, that is weight, height, number, etc. W0 or W0 is the initial size at the beginning of the period. R is the growth rate, that is also relative growth rate. T is the time of growth and E is the base of natural logarithm. W1 equal to W0 ERT. Next, here R is relative growth rate. It is also the measure of the ability of a plant to produce new plant material, efficiency index. Hence, final size W1 depends on initial size W0. Quantitative comparisons between the growth can also be made in two ways: absolute growth phase and relative growth phase. Absolute growth rate means measurement and comparison of total growth per unit time. That is called absolute growth rate. Relative growth rate means measurement of growth of the given system per unit time expressed on a common basis, example per unit initial parameter. Here you can see diagrammatic comparison of absolute and relative growth rates. Both leaf A and B have increased their area by 5 cm square in a given time to produce A1 and B1 leaves. Here coming to conditions or essential element for growth. First of all, water. For cell enlargement, turgidity of cell helps in extension growth. Water provides medium for enzymatic activities needed for the growth. Oxygen, it helps to release metabolic energy for growth. Nutrients, macro and micro elements are needed for the synthesis of protoplasm and act as a source of energy. Temperature, plants have an optimum temperature at which growth is maximum. Deviation from this range could be detrimental to its survival. Light and gravity affect certain phases or stages of growth. So these are the conditions or essential elements for plant growth: water, oxygen, nutrients, temperature, light, gravity. Now coming to differentiation, dedifferentiation and redifferentiation. Differentiation, you are familiar with this term. You will get this term in class 9. Now listen. Differentiation is a process in which the meristem cells that are root apical and shoot apical and cambium differentiate and mature to perform specific functions. In this, cell walls and protoplasm undergoes structural changes. Capacity of cell division is lost. Example, loss of protoplasm to form a tertiary element. They also develop very strong, elastic, lignocellulosic secondary cell wall to carry water to long distance even under extreme tension. As you can see, simple cell, then meristematic cell divides to root cell, then leaf cell, and then coming to sheath cells. Okay. Under certain conditions, living differentiated cells regain the capacity of division. This is called dedifferentiation. Example, formation of meristems, interfascicular cambium and cork cambium from differentiated parenchyma cells. Dedifferentiated cells can produce cells, then again lose the capacity to divide but mature to perform specific functions. It is called redifferentiation. Okay, or also called transdifferentiation. Plant growth is open, that is, it can be indeterminate or determinate. Differentiation is also open because cells or tissue arising out of the same meristem have different structure at maturity. Final structure at maturity of cell or tissue is also determined by the location of the cell. Example, cells positioned away from the root apical meristem differentiate as root cap cells, while those pushed to periphery mature as epidermis. You can see this diagram. Differentiation, then it converted into dedifferentiation, stem cells like state, transient phase, then it goes to redifferentiation or transdifferentiation, or it goes to cell death, or it goes to cell cycle. Okay. Now, plant development. It is a process that includes all changes in the life cycle of an organism from seed germination to senescence. What is the plant development? Means it is a process that includes all changes in the life cycle of an organism from seed germination to senescence. Senescence means, senescence in plants refers to the process of aging and eventual death of plant tissue, organs or the entire plant. That is called senescence stage. It is the sum of growth and differentiation. It represents below. You can see meristematic cell, then plasmatic growth, differentiation, then it goes to mature cell, then senescence came and death occurred. Okay. Here also zygote stage, 16-celled embryo, heart-shaped embryo, then juvenile stage, then coming to adult stage, and then reproductive stage. Now, plants follow different pathway in response to environment or phases of life to form different kind of structure. This ability is called plasticity. Remember the term. Plants follow different pathways in response to environment or phases of life to form different kind of structure. This ability is called plasticity. Example, heterophyly due to phases of life. Example, in cotton, coriander and larkspur, the leaves of the juvenile plant and mature plant are different in shape. See the diagram? Juvenile phase, the leaf structure is different, while in the adult, the leaf structure is different. Heterophyly due to environment. Example, difference in shape of leaf produced in air and water. Example, buttercup. You can see the B diagram, buttercup, where the in normal terrestrial habitat the leaf shape is different, while in the water habitat the leaf shape is different. Okay, this is called plasticity. Means they respond to the environment and they change to different structures. That is called plasticity. Now plant development. Factors controlling plant development. Intrinsic factor, extrinsic factor. Coming to intrinsic factor, intracellular, that is genetic. Intercellular, example, plant growth regulators. Means different hormones. This is the intrinsic factor. Coming to extrinsic factor, light, temperature, water, oxygen, nutrition. These are the extrinsic factors. Okay. Now, plant growth regulators. Plant hormones or phytohormones. Plant growth regulators PGR are small, simple molecules that regulate plant growth. Based on the functions, PGR are two groups. One is plant growth promoters and plant growth inhibitors. Plant growth promoters means for growth promoting activities like cell division and enlargement, tropic growth, pattern formation, flowering, fruiting and seed formation. Example, auxins, gibberellins and cytokinins. Plant growth inhibitors means for growth inhibiting activity. Inhibit means rokna. Okay. For growth inhibiting activities like dormancy. Dormancy means dormancy in plants refers to a state of temporary metabolic inactivity or minimal activity characterized by a reduction in growth and development. That is called dormancy. And abscission. Abscission in plant refers to the natural process of separation or shedding of a plant part such as leaf, flowers or fruit from the plant body. That is called abscission. So, plant growth inhibitors means for growth inhibiting activities like dormancy and abscission respond to wounds and stress of biotic and abiotic origin. Example, abscisic acid and ethylene. So there are 5 plant growth regulators. Auxin, gibberellin and cytokinin are plant growth promoters. They promote the plant growth, while abscisic acid and ethylene are plant growth inhibitors. Ethylene fits either of the group, but it is largely a growth inhibitor. Next, first of all, auxins. Auxin comes from Greek word auxein, that means to grow. Charles Darwin and his son Francis Darwin observed that the coleoptiles of canary grass responded to unilateral illumination by growing towards the light source, phototropism. It was concluded that the tip of coleoptile caused the bending of the entire coleoptile. F.W. Went isolated auxin from the tip of coleoptile of oat seedlings. Auxin was first isolated from human urine. Auxins are produced by growing apex of stem and roots from where they migrate to regions of their axons. So, auxins, there are 2 types of auxins, natural and synthetic. Natural examples are indole 3-acetic acid IAA, indole butyric acid IBA. They are isolated from plant, that's why they are called natural. Synthetic example NAA, naphthalene acetic acid and 2,4-D, 2,4 dichlorophenoxyacetic acid. Okay, 2,4-D. These are synthetic auxins. Function, they initiate rooting in stem cutting for plant propagation. If the stem is cut, then they propagate the rooting process of the stem. Promote flowering, example in pineapples. Third, to prevent fruit and leaf drop at early stage. Fourth, promote the abscission of older leaves and fruits. Fifth, induce parthenocarpy, example in tomatoes. Sixth, they are used as herbicides. 2,4-D is used to kill dicot weed. It does not affect monocot plants. It is used to prepare weed-free lawns. 7. Controls xylem differentiation and helps in cell division. These are the functions of auxin. What they work. Here the word in 6th point given. These are also used as herbicides. Auxin herbicides induces the overproduction of abscisic acid, which involved in stress response, dormancy and senescence. It also causes ethylene burst, which leads to the growth abnormalities and aging in plant, and finally the plant dies. Simple. That's why they are used as herbicides. Remember, synthetic auxins are only used as herbicides. Okay. Here some more points. In higher plants, the growing apical bud inhibits the growth of lateral or axillary bud. It is known as apical dominance. So auxin are also helped in apical dominance. Apical dominance means the growing apical bud inhibit the growth of lateral bud. That is called apical dominance. Removal of shoot tip, that is called decapitation, result in the growth of lateral bud. It is applied in tea plantation or hedge making, etc. Here you can see apical dominance in the first diagram, and it is cut, apical bud is removed and lateral branches are formed. Okay, that is called decapitation. Removal of the shoot tip is called decapitation. Okay. Second is gibberellins. These are acidic PGR. Gibberellic acid, okay, that's why they are called acidic PGR, plant growth regulators. E. Kurosawa treated the sterile filtrate of Gibberella fusicori, a fungus that causes Bacane disease or foolish seedling in rice, to healthy rice seedlings. As a result, it showed that the symptoms of Bacane disease. Later, the active substance were identified as gibberellic acid. There are more than 100 gibberellins are there, GA1, GA2, GA3 and etc. and so on in fungi and higher plants. Gibberellic acid GA3 or terpenes is one of the first discovered and most intensively studied gibberellins. Function. They cause increase in length of axis, so they are used to increase the length of grape stalks. Second, to elongate and improve the shape of fruit such as apple. Third, they delay senescence, so the fruit can be left on the tree to extend the market period. Fourth, GA3 is used to speed up malting process in brewing industries. These are the functions of gibberellins. Some more points. Fifth, sugarcane stores sugar in stems. Spraying sugarcane crop with gibberellin increases the length of the stem. It increases the yield by as much as 20 tons per acre. Spraying juvenile conifers with gibberellic acids hastens the maturity period. It leads to yearly seed production. 7. For bolting. Bolting means internode elongation just prior to flowering in beet, cabbage and many plants with rosette habit. What do we mean by rosette habit? Now, rosette habit refers to a type of plant growth pattern where leaves are arranged in a circular or flat spreading pattern from a short stem or axis at ground level. That is called rosette habit. Third is cytokinins. Epps, Cook and co-worker observed that from internodal segments of tobacco stem, the callus, Callus means a mass of undifferentiated cells proliferated only if the nutrient medium was supplied with extract of vascular tissue, yeast extract, coconut milk or DNA. Skoog and Miller later identified and crystallized the active substance and termed as kinetin. Cytokinin were discovered as kinetin, N6 porphyrylaminopurin, an adenine substrate derivative from the autoclaved herring sperm DNA. What do you mean by autoclaved herring sperm DNA? Now it refers to the DNA extracted from the sperm of herring fish and subsequently sterilized using steam at high pressure. That is called autoclaved herring sperm DNA. Kinetin does not occur naturally in plants. Next, gelatin from corn kernels and coconut milk is the natural substance with cytokinin-like activities. There are some synthetic compounds with cell division promoting activity. Natural cytokinin are synthesized in regions of rapid cell division, root apices, shoot buds, young fruits, etc. Functions of cytokinin are they play a role in cytokinesis. They help to produce new leaves, chloroplast in leaves, later shoot growth and adventitious shoot formation. They help overcome the apical dominance. They promote nutrient mobilization, which helps in the delay of leaf senescence. Fourth is your ethylene, C2H4. Cousins confirmed the release of a volatile substance from ripened oranges that hastened the ripening of stored bananas. Later, this substance was identified as ethylene. Ethylene is a simple gaseous plant growth regulator. It is synthesized in large amount by tissues undergoing senescence and ripened fruits. Functions: it influences horizontal growth of seedling, swelling of axis and apical hook formation in the dicot seed. You can see apical hook formation. It promotes senescence and abscission of plant organs, especially of leaves and flowers. It promotes fruit ripening. It enhances respiration rate during fruit ripening, that is also called respiratory climactic. It breaks seed and bud dormancy, initiates germination in peanut seed, sprouting of potato tuber. Some more functions: promotes rapid internode or petiole elongation in deep water rice plants. It helps leaves or upper part of the shoot to remain above water. Promotes root growth and root hair formation. This increases absorption surface. 7th, used to initiate flowering and for synchronizing fruit set in pineapples. It also induces flowering in mango. 8th, it is widely used in agriculture. The most widely used source of ethylene is ethiphone. Ethiphone is an aqueous solution, is readily absorbed and transported within the plant and releases ethylene slowly. Ethiphone hastens fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits. Thinning of cotton, cherry, walnuts. It promotes female flowers in cucumber, thereby increasing the yield. Now coming to 5th one, abscisic acid, ABA. During mid-1960s, it was reported 3 kinds of inhibitors: inhibitor B, abscisic second and dormin. They were chemically identical and now known as abscisic acid. ABA is the derivative of carotenoids. It regulates abscission and dormancy. Functions: it acts as an inhibitor of plant growth and metabolism. It inhibits the seed germination. It stimulates closure of stomata in the epidermis. It increases the tolerance of plant to various kinds of stresses. Therefore, it is also called stress hormone. It is an important role in seed development, maturation and dormancy. Seed dormancy by ABA helps to withstand desiccation and other factors unfavorable for growth. Desiccation means the process of becoming completely dried. Okay. Interaction of PGRs. PGRs play individualist or synergistic role. Such role may be complementary or antagonistic. Antagonistic means one that contend with or oppose another. PGRs interact to affect dormancy in seeds or buds, abscission, flowering, senescence, vernalization, apical dominance, seed germination, plant movements, etc. Vernalization means the artificial exposure of plant or seeds to low temperatures in order to stimulate flowering or to enhance seed production. In most cases, ABA act as an antagonist to gibberellic acids. Opposite. Okay, antagonist means I told you it means one that contains with or oppose another. So it opposes the gibberellic acid. Now coming to factors influencing the action of PGR. Intrinsic factor, genomic control, we all go through it. Extrinsic factor, light, temperature. First of all, role of light and temperature on flowering. First is photoperiodism. It is the response of plants to periods of day and night. Some plants require light to induce flowering. Based on light duration, plants are three groups. Long day plant, they require the exposure to light for a period exceeding a well-defined critical duration. Short day plant, they require the exposure to light for a period less than the critical duration before the flowering is initiated in them. And day neutral plants, they have no correlation between exposure to light duration and induction of flowering. While shoot apices modified into flowering apices, they cannot perceive photoperiods, the site of perception of light in dark duration in the leaves. It is a hypothesis that there is a hormones for flowering. When plants get enough photoperiod, the hormone migrate from leaves to shoot apices to induce flowering. Next is vernalization. I told you it is the phenomenon in which some plants depend quantitatively or qualitatively on exposure to low temperature for flowering. Advantages: it prevents precocious reproductive development late in the growing season. Precocious means ripening before the natural time. It enables the plant to have sufficient time to reach maturity. Example: some food plant, wheat, barley, rye have two varieties: spring variety, winter varieties. Spring varieties, these are normally planted in the spring and comes to flower and produce grain before the end of the growing season. Winter variety, if the planted in spring, would normally fail to flower or produce mature grain within a span of a flowering season. Hence, they are planted in autumn. They germinate and over winter come out as small seedlings, resume growth in the spring and are harvested usually around midsummer. Vernalization in biennial plants. Biennial are monocarpic plants that normalize flowers and die in the second season. Example: sugar beet, cabbage, carrots, etc. Subjecting the growing of biennial plant to cold treatment stimulates a subsequent photoperiodic flowering response. This is the vernalization. And with this, we have completed the whole chapter, and the important points of NCERT are also covered in these lines of this slide of the PDF, as you can see, and go through your NCERT line by line, and also watching this video. If you found any doubt, simply contact me. My contact number is in the description of the video, and like this video, comment and subscribe to my channel if you are new. So thank you. Jai Jagannath.